Hey everyone, so today we're going to finish chapter 10, microbial metabolism, and this is part 4, biosynthesis and the crossing pathways of metabolism. So in this section, we're going to learn about how we can use some of the components that we get during the breakdown of glucose and the processes that we've already talked about, mostly in section 3, and how we can use those to build new molecules. Because the goal of the cell is to divide and grow, and so this is the way that their cells are able to pull out some of the building blocks that they need as they're going through these processes. So this gets us to the term amphibolism, and amphibolism means amphi means two or both, and so that means metabolism that's going in both directions. So what that means is that when things are being broken down or catabolic reactions, they're coupled or paired with anabolic reactions. And many of the pathways, like if we're looking at glycolysis, if we get to pyruvate, the pyruvate doesn't have to go into the Krebs cycle. The pyruvate can be removed from that reaction and then used as a building block for other components. And we'll see that for other things as well. So here's the overall pathway. This should be familiar to you now because we've spent a bit of time looking at it. So as you can see here, notice the direction of the arrows. So you can see there's different places that things can enter into these pathways to break down to generate ATP, but there's also intermediates that can be pulled out in order to build amino acids, carbohydrates, and lipids. And so we can see these arrows going in both directions. And that's the amphibolic nature of these pathways. So the precursor molecules, this is what can be used as a source to build other compounds. And here are the three big ones that we see in the whole glycolysis, transition, and Krebs cycle pathways that we've talked about already. So the intermediate glyceraldehyde 3-phosphate, it can be diverted from glycolysis and converted into the precursors to make carbohydrates, triglycerides, and amino acids, which of course are the building blocks of proteins. Pyruvate provides intermediates to build amino acids. And also we can use pyruvate as a starting point to make glucose, and that is called gluconeogenesis. Great big old long word, and really all it means is making glucose. So using pyruvate just to make glucose. If you start to run out of that sugar, the cell can make more. And then acetyl-CoA is also a precursor and can be converted into several amino acids and condensed into a lipid formation. So these molecules serve as precursors for nucleic acids, the pathways to synthesize purines and pyrimidines, which of course are the A's and the T's and the C's and the G's, or the U's if you're talking about RNA. And the pathways to make amino acids. These can be dependent upon intermediates through the Krebs cycle, so components of the Krebs cycle can be removed again in order to make these precursors. And if you are in carbohydrate deprivation, if the cell is starving, is really what that's saying, you can actually convert amino acids and use them as the energy for as the energy in the Krebs cycle. Now that would not be an ideal situation. When you start using and metabolizing proteins or amino acids for energy, that's not a good condition for the cell to be in, and they would rather look for other sources of nutrients like carbohydrates, especially if they're metabolizing their own proteins. So a crucial role in glucose metabolism is biosynthesis through all the different pathways that we've looked at. Building cell walls, using carbohydrates to build cell walls, whether it's cellulose if it's a plant cell, or the carbohydrates to go into other cell walls and to form glycogen storage units. Amino acids or proteins rather are a large portion of the cell's constituents, and they need to be made. Um, E. coli can make all 20 amino acids. None of them are essential, so all of them, they have biosynthetic pathways that they can use. And of course, these are the building blocks to make proteins. So again, the cells can pull intermediates out of the process, like pyruvate, and use that as a building block to make the proteins. Once you've built your proteins and your nucleic acids and your lipids, and you've got sufficient glycogen and sugar storage, then you can and make enough ribosomes, etc. Then you can double your size and divide your cell, and now you've made a second cell. And so that's the whole point of building all these things, is to make more cells. So now we're at the end of this section. You should be able to answer these 3 quick questions, and I will see you next time.